Good morning, everyone, to one and all present here. It's our pleasure to welcome you all to one-day hands-on workshop on quantitative PCR from gene expression to data interpretation. Without further ado, I would like to invite Professor R. Krishna, Head of Department of Bioinformatics, to deliver the welcome address.
Respected Honorable Vice Chancellor Professor Prakash Babu, respected Dean, School of Life Sciences, Professor Joseph Selvin, Ajay from Application Scientist from BioRod, Professor Dinakar, Coordinator of this program, all aides of the university, faculties, research scholars and students. It's really very happy and immense pleasure to give the welcome address on the occasion of one-day hands-on workshops on quantitative PCR from gene expression to data interpretations that is conducted by the Department of Bioinformatics, Pondicherry University. Department of Bioinformatics is one of the best departments in the country. We know that it has recognized as a center of excellence in 2007 itself. And we are running the four independent programs, and this is the first department NEP program we started, that is UG NEP programs. And we have 250 students. Along with that, we have 12 faculties, all are very interdisciplinary. It's the best department in the country that we need. And it is a great pleasure that and honor inviting our Honorable Vice Chancellor for this conference to inaugurate the workshop. So thank you very much. You are always very supportive to the department, and also very thank you that you are giving a presidential address in this conference, that workshop. I also welcome our Dean, School of Life Sciences, for giving the felicitation address in this conference. Thank you, sir. I also thank you for welcome our coordinator, Professor Dinakar Rao, Department of Bioinformatics. I also welcome all aides of the departments, research scholars and students. Thank you very much. That indeed.
Now I invite Professor A. Dinakar Rao, Coordinator, Department of Bioinformatics, to speak on the scopes and objective of this workshop.
Dignitaries on the dais, our beloved Vice Chancellor, Professor P. Prakash Babu sir, and Dean, School of Life Sciences, Professor Joseph Selvin sir, and our HOD, Professor Krishna and Dr. Murli, and our senior professor, PMOD awardee, Professor Chandra, and HODs of the department, other departments, Dr. Thiru, Dr. Prashant, and Dr. Madam Rachel, and my colleagues, and students from KV. Good morning. So the scope of this seminar, as you know, PCR, which is known as polymerase chain reaction, is a technique we use to amplify a segment of DNA or RNA. Now, I would like to thank COVID-19 for posing many challenges to us and introducing many things to us, like mask, sanitization, and then like RT-PCR test. Okay, during that time everybody knows about RT-PCR. RT-PCR test. Okay, RT-PCR is nothing but real-time PCR, which is more accurate, sensitive, and has very wide applications from biological sciences to industry. You can see that one. So there is two main reasons for this workshop. One is inviting KV students, biology students, and showing our laboratory facilities, department facilities, and encouraging them to take their higher education in biological discipline. And second reason is to update knowledge in our research students, School of Life Sciences, on real-time PCR, how to use the real-time PCR and how to analyze the real-time PCR data and to apply in their research. So biorad technician, biorad engineer is called, invited, and biorad person is giving lectures on real-time PCR, how to set up the reactions and how to calculate the data and how to interpret your results. So, with this intention, when I contact our HOD, he readily accepted to support logistic support, and in this regard, I would like to thank our HOD for your support. Thank you, Dr. Krishna. And also, I would like to thank Mr. Anbal again, who is follow up this paper and made the things done, our office personnel. And I would like to invite, ask research scholars, please try to utilize real-time PCR in your research, and it will be added technology, added strength in your research. Also, it will, provide strong support to your publication to publish in higher impact journals. And I would like to give big applause to our Honorable Vice Chancellor for his commitment and for his support and setting good example in the campus to our faculty by taking, is taking one neuroscience paper, full paper to as a soft core paper in School of Life Sciences in the Department of Bioinformatics. Thank you very much, sir, for setting a good example in the department. I hope you are the first person who is taking the full paper in this university as Vice Chancellor. Thank you very much, sir. And I would like to conclude. So this PCR workshop is arranged to have, knowledge on real-time PCR, how to use real-time PCR for your research. So please use this workshop to enhance your knowledge, how to use your research, and how to use PCR method in your research for your thesis work. Thank you very much for this opportunity. Thank you.
Thank you, sir. I now invite Professor Joseph Salvin, Dean, School of Life Science, Pondicherry University, to deliver the inaugural address.
Good morning, all. Very esteemed and respected Honorable Vice Chancellor, Professor Prakash Babu, and respected Head of the Department, R. Krishna, and Professor Tinagara Rao, organizer of this event, Dr. Murali, and respected colleagues, particularly our senior professor, Professor Chandrasekharan. Very happy to see him. Welcome you, sir. So, and respected colleagues as well as dear students. This is one of the unique moment. I am not taking much time. I wanted to say a few words about the evolution of PCR. Now we are reached the droplet digital PCR or high throughput digital PCR. So previously we usually amplify one gene. Now we are able to amplify thousands of genes at a time. So that is the developments are going on. So thereby this is nowadays called high throughput. Usually we use the students should understand anything we are doing in the lab, like one or two experiments, the same experiment robotically we make it thousands and lakhs together, and it is high throughput platforms. So you should understand the basics of PCR always. Usually I ask some of the students the question, what's the difference of primer and probe? Both are short oligonucleotide, right, targeting the DNA. And what's the difference? Primer always starts from 3' and 5'. Then probe will be any part of the target DNA. Then what is the design? What is the requirement of the design? The design always required, some basic, actually, 3 domains of our archaea, bacteria, and eukarya. you should understand there only it starts A plus C, AT and GC content. So GC means always triple bond and AT means double bond. So GC is very important, guanine and cytosine. So that will decide the primer design or probe design. Now synthetic biology, maybe our Professor Chandrasekharan is here. So it will give another lift in the School of Life Sciences about synthetic biology. So all these small short oligonucleotide designs required, you should have the knowledge of TM value. TM value means melting temperature. DNA melting means double stranded DNA is denaturation. So all these basic, hopefully you will understand from this one day workshop. Lot of things because it is very advanced. At the time of COVID, we are running here and there. So many people trained, they are ready. So India can face any situation of outbreak kind of situations. We cannot escape because of these are all naturally happening. So with this, I'm joining with Professor Dinakara Rao. We are changing the history of Pondicherry University. Our Professor, Honorable Vice Chancellor, now joined with teaching. So this is the fashion and legacy of teaching. So that means sir is together with us. So it's a life sciences with Honorable Vice Chancellor, Professor Chandrasekharan. We are going in another direction. So I wish you all good luck. You have a pleasant day. Definitely have a strong deliberation. Enjoy it. Thank you.
We are honored to have with us Professor P. Prakash Babu, Vice Chancellor of Pondicherry University, to deliver the presidential address.
Very good morning, all of you. Dignitaries on the dais, Professor Selvin, Dean, School of Life Sciences, Professor Krishna, Head of the Department of Bioinformatics, other colleagues, Professor Dinakar Rao, Coordinator of the program, Dr. Murali, Professor Chandrasekharan, Heads of the Departments of Life Sciences, other faculty colleagues sitting, students, research scholars, and the KB students. So as they said that I joined teaching, yeah, I joined teaching because teaching is my passion. And for last, I think I have completed 10 months, so I could not stay without teaching, therefore I joined it. It's like a habit. We are habitual teachers, so we have to make it someday. And also going to the different levels is actually also my passion. Actually for the science day, I choose some school in the rural area and I go in the month of February because we have to deliver the requirements where it is required. So it's out of my passion I do things, and I think Biorad people are here and they are helping us. The first instrument I purchased was Biorad Power Pack. So I got one DST Young Scientist project. My first project in my career, when I soon after the 1 year I just joined as a lecturer at that time. So that worked actually for several years. In fact, it was very beautiful piece. Still it is there. Of course, it's not working. I don't want to throw it also. Still it is there in my lab. And KV students, it's a great opportunity for you. I hope your teachers also here. Who are the teachers? Here? Okay. Very good. I think you should involve biology teachers also. So the reason, first time in my life or career or student life, so we keep reading about the cell biology. We have one subject in BSc and of course intermediate also. We used to read this cell structure and also cell organelles and all. So how many of you know the cell? KV students. Cell, you know, animal cell, okay, right. Have you seen the cell? How many of you have seen the cell? Anybody? Not on the books, real cell. So I saw that cell during my PhD. When I joined for PhD, so we used to have one microscope, that's I think Nikon microscope. I saw first time the cell in my life. So I got so fascinated by that. So now the KV students, now you are in +1, +2. You are able to see. That's what Professor Dinakara was indicating to me. we showed them how the gene can be amplified, what are the instruments and all, what is their function. You can see the protein. So when we run the SDS page gel, first time I saw that, oh, these are the protein bands. So how the protein will be look like. So till then we do not know. So you are actually, Kendriya Vidyalaya students of this Pondicherry University are very lucky that you are in the part of the one of the best central universities in the country. Since you are part of this one, you should make use of the presence, especially teachers and the principal. You should make use of the presence of the students in the Pondicherry University and keep them to visit several departments. So you should get exposure for all the departments, then you choose your career. Visit the physics department, visit the chemistry department, okay, so all the departments, then you will have a idea what you should do in your career. And this is a great opportunity for you to be part of it. As earlier speakers indicated, I think Professor Selvin, he gave a historical evaluation of the PCR, and I think as the students, I think as Professor Dinakar indicated that the COVID taught us several lessons. COVID taught us several lessons. So before COVID, we do not know, even I do not know how to teach online. So COVID made me to learn how to deliver an online talk using the PowerPoint presentation. Of course, we used to teach to the computers because no students were there. Though it's only technically they will be there online, but nobody will be there. Okay, so that's fine. So apart from that, we also learned the health emergencies. The world, it's not the country, entire world learned the health emergencies can occur anytime, anytime. That's why even the government of India or the education department is also encouraging faculty members to take some classes online, so that let us be prepared for such a kind of eventuality. As you know, the human evolution, so we teach, when we teach some of the diseases, we take for example malaria. Now everybody knows, student knows, children knows malaria, about malaria, and malaria is not yet vanished. Malaria still it is there, right? In the early 70s, Indian government started a program called Malaria Eradication Program. Then after 10 years, in 1982, government realized that malaria cannot be eradicated, so they changed the name to Malaria Control Program. So what I would like to impress upon you is, every 10 years, these pathogens or infectious agent, they come with a breakthrough, because their genetic evolution is such that they are more intelligent than the human beings. Yes or no? They are more intelligent than human beings because they are born much earlier to us. In the evolution, if you take the unicellular organisms or the viruses and bacteria, they are born earlier to us. So they can manipulate or they can change their genetic phenomenon so that they can become a resistant. It's called drug resistance, as most of you know that one. That's why, so during COVID, many people used medicines, drugs indiscriminately. Indiscriminately. We have used Dolo 50, Dolo 650. Many people who have used it. If there is a fever, use it. So every drug, even antibiotics, people use all kinds of antibiotics because nobody knows, even the doctors, they do not know how to respond. Only problem is, only importance is that let us save the life. Use several drugs, and so that caused the drug resistance. In the Nagpur, one of the hospitals, they made a wonderful study. So during COVID time, they collected the drainage samples. Drainage samples, they found that so many drugs are there in the drainage. That means they are all human consumption and they are eliminated. So once these drugs are actually in the general environment, so environment will become drug resistant. So all the bacteria, viruses that they are in the environment will become drug resistant. Drug resistant now. So all these things again, when we come back to this one, RT-PCR plays a very vital role. If you see the very small sample, so as children you must be knowing, you are there during COVID now. Okay, so have you realized that there is a COVID and all? You understand that. So anybody has done throat swab test? Yeah. So throat swab, when they put the cotton swab and take out the small mucus, very mucus, how much sample will get? From the small, it's negligible, you cannot see with the naked eye, that is being used to check the RT-PCR, to test whether you have a positive or negative. You can imagine, now you know the importance of RT-PCR, right? So every disease, RT-PCR plays a very crucial role. The ultimate test is the RT, because you can have the small piece of sample without even knowing that one. So that is good enough to know, and also this is a kind of final test. Final test, okay, this is a kind of confirmation test. So this RT-PCR in the life of the scientist plays a very crucial role for the research. You can see the gene amplification. By amplifying the gene, you can see, if you take one particular disease like a cancer, by studying this one, you can see that one, what are the genes are getting expressed. So my students, they do with a brain research, brain tumors. There is a tumor, this is called as glioma, deadliest tumor. If anybody can find any drug against that glioma, they will get Nobel Prize. So it gives only after disease is being seen or identified by a pathologist, it will not give more than 14 months, 13 to 14 months, that's all. There is no drug at all. So we take the students, they take the small sample and check for what are the genes are getting amplified. Okay, increased or decreased. So how the genetic manipulation or the derailment of the genetic mechanism is going on in these small samples can be studied by using this one. So when I'm giving one or two examples, because the importance of the RT-PCR, especially in the health and disease, that plays an very important role. So tomorrow, if you want to become a scientist, I think this is the best area to choose and use the RT-PCR. Now you are lucky to have the getting exposed to the seeing that instrument and working with that instrument is amazing for you. So I compliment the Department of Bioinformatics. So I think we should keep on doing the entire university. Now today we have a heads and deans meeting where I am going to announce or inform the heads and teams, since we have completed 40 years of existence of this university, we should take up this as an opportunity to see that the many more activities are done in the university across the disciplines, across the departments, and involve the school students to the college students and the university students. So it's throughout that, let us campaign as a science or a humanities or social sciences across the Pondicherry or the Tamil Nadu or in the entire country. Let us do as many activities as possible. So with this, I once again thank the department for taking initiative for everything. Bioinformatics is actually going in a forefront to conduct certain activities of the academic and research. I compliment and I thank for all these things arranged, and also Dean and other people who are sitting here, and media people are always be there to take the mission in forefront, and that's what today I was indicating to the guests that they can come. So we have a media department which is very systematically they do. All these programs will be uploaded by evening in the university website. So see, whatever you do, even the people around the globe also should know what Pondicherry University is doing it. So let us thank them also in every place wherever they are. And I wish such a program should continue, and we will celebrate the National Science Day in a very big way by involving all the schools and departments of especially the sciences. So thank you very much, and I wish you all best of luck.
Thank you, sir, for your motivating and thought-provoking speech. I now invite Dr. A. Murali, Department of Bioinformatics, to deliver vote of thanks.
Dietaries on dais, off dais, ladies and gentlemen, good morning. It's my pleasure to present the vote of thanks on behalf of the Department of Bioinformatics, Pondicherry University. We are very grateful to have our Professor, Honorable Vice Chancellor Professor Prakash Babu sir, for his gracious presence, for spending and delivering a presidential lecture today. And to be in brief, Professor Prakash Babu sir is not an academician and administrator, he is a good teacher also, as was proven by his intention to take a class. And for those who has not attended the class, you might have seen his teaching ability in explaining you the PCR in very simple words. And despite of your busy schedule for being here and delivering the talk, I thank you very much, sir. Thank Mr. Ajay Deepak Bhaskaran, a senior field application specialist from BioRed, who is going to run the show in related to the talks and the hands-on experience on PCR. Thanks for being here. And I would like to thank Srimati Jasmine, Principal, PM Sri Kendriya Vidyalaya number 2 Pondicherry, and the biology teacher for allowing the young minds to participate in this program and making the future ready scientists. And I also thank the AR transport section for providing the transport for the KV students to from the KV to the bioinformatics department. I would like to place my heartfelt thanks to my colleagues from bioinformatics department as well as the sister departments in the life sciences who graced their presence for successful organization of this event. More important, for a successful organization of any such event, volunteers are required, and the volunteer extended by the research scholars and students is a key point for the successful organization of this event, and I thank them for all, one and each. The backbone of success of any organization is the non-teaching staff, the technical staff, and the media, and I thank each individual for their active participation in this event. Quite overlooked, special thanks to the outsourcing persons who kept this premises very clean, and they deserve a special thanks for cleanliness in the campus around. And no event is successful without having a participants around, and I thank all the participants for showing enormous interest in participating, especially the young minds. I'm pretty sure you will carry fruitful memories with you when you go back, and also a lot of hopes. And I thank you for being here. Literally, it is impossible to list each and everyone who contributed to the successful event, and listed or unlisted, whoever contributed takes my thanks for the event organization. And I thank you one and all. Thank you.